Good evening, everyone, and welcome to the 2021 Your Health Lecture Series at Calvin University. My name is Arlene Hogewerf, and I am the Dean of the School of STEM and Health Sciences. I'm delighted to have our audience here in person, as well as those who are joining us online. The Your Health Lecture Series is a unique educational partnership between Calvin University, the Michigan State University College of Human Medicine, and Spectrum Health. The goal of this series is to feature researchers who can speak to a wide variety of topics for a public audience. The research scientist or clinical researcher comes to Calvin and meets with pre-med students prior to this evening public lecture. Additionally, Calvin pre-medical students have the opportunity to have a roundtable discussion with Michigan State University College of Human Medicine students. So we are delighted to have this partnership. This is our 8th year, and this partnership allows our students to have access to both of these groups, and as well as providing a service to the community. So once again, welcome. I want to now introduce Mark Briebe from the Michigan State University College of Human Medicine, and he will be introducing our speaker this evening.
All right, thank you, Arlene. Thank you for that kind introduction. Like Arlene said, my name is Mark Briebe. I'm the Director of Community and Government Relations at Michigan State University College of Human Medicine. I just want to say it's such a pleasure to be back at Calvin University. As Arlene said, we've done this for about 8 different sessions. And I personally enjoy coming back because I graduated from Calvin in 2004. I was married in this chapel in 2005. So this event always is very special to me. I want to thank Calvin for hosting this lecture. I also want to thank Spectrum Health for co-hosting the event. I did want to mention we do have several lectures scheduled in the coming months. All will also have a virtual option. On Wednesday, October 13, we have Dr. Zakia Alavi on depression and suicide prevention. Monday, October 25, we have Dr. Mona Hanna-Atisha on What the Eyes Don't See, a story of crisis, resistance, and hope in an American city. Dr. Mona was the pediatrician in Flint that helped find out about the lead crisis in Flint. And then on Wednesday, December 1, we have Dr. Cameron Risma. He'll actually be over at Grand Valley State University, and he's speaking on substance use disorder and mental health. So with that being said, I would now like to introduce our speaker this evening. Dr. Rebecca Nickmeyer received her PhD from the University of Cambridge and completed postdoctoral training at the University of North Carolina at Chapel Hill. She is currently an associate professor in the Department of Pediatrics and Human Development at Michigan State University and co-director of MSU's Center for Research in Autism, Intellectual, and Other Neurodevelopmental Disabilities. Her research addresses genetic influences on infant brain development, sexual differentiation of the human brain, and the microbiome gut-brain axis. It was recently announced that Dr. Nickmeyer is leading the largest of its kind effort to understand the impact of genetics on early brain development and neurological disorders such as autism, schizophrenia, and attention deficit hyperactivity disorder. Supported by a $5.5 million grant from the National Institutes of Health, the project seeks to illuminate which genes steer the course of brain development in both healthy and neurodivergent children. This work could help researchers develop new treatments for disorders at a much earlier stage. Please help me welcome Dr. Rebecca Nickmeyer.
Thank you very much for that kind introduction, Mark. I'm very excited to be here speaking with you all today. And today I'll just be speaking on one aspect of my research, which is the microbiome gut-brain axis. So we'll be talking about babies, we'll be talking about brains, and the intriguing ways that bacteria and other microbes in our gut may shape our lifelong mental health. I'm going to start just with a quick disclosure slide. I think this is always good. So I'm not talking about anything related to this today, but I do serve on the Professional Advisory Board of the Turner Syndrome Society of the United States, unpaid, and the Scientific Advisory Board of the Turner Syndrome Research Network. In the past 2 years, I've received grant support from the National Institutes of Health, as well as the Foundation of Hope for the prevention and treatment of mental illness. And I also serve as a consultant on a grant that was funded by Nestle and Wyeth, which is on the role of nutrition in the microbiome and brain development, but I'm not going to be showing any of that data today. So this is an overview of what the talk's going to look like. We're going to start out by talking about what is the gut microbiome. Then we'll talk about the effects of the gut microbiome on brain and behavior in animals briefly, what we know about the effects of the gut microbiome on brain and behavior in humans. Most of the talk is going to focus on my own research into the importance of infancy, both in the development of the gut microbiome and the brain, and how they talk to each other. Which brings us to the next section, which would be how does the microbiome communicate with the brain? For a microbe, that's a long distance to cross. And finally, we'll talk about the clinical implications of this work. What could this really mean for patients and families and the general public? But I want to start by asking 2 questions, and I will ask them again at the end. We'll see if there's any change or not. So my first question is, do you think that manipulating the gut microbiota is a promising strategy for supporting cognitive development? You could raise your hands or not. And the second question is, do you think that manipulating the gut microbiota is a promising strategy for treating and preventing psychiatric disorders? So, related but slightly different question. All right, so we'll see if opinions change or not. All right, so what is the microbiome? So a microbiome is a community of microorganisms living in a particular environment. That could be in a hot thermal pool, it could be in your armpit. There we go. We think of these organisms as being commensal, so they're living in close association. Many of them have symbiotic relationships, both with each other and with the host, if it's a microbiome on the body. So that means they're mutually beneficial, they help each other out. But they can also be competitive, both with each other, competing over resources in that environmental niche, and also potentially fighting with the host for access to things like nutrients. And finally, some of them will be actually pathogenic, so capable of causing disease. And also the term microbiome is also used to refer to the combined genetic material of the microorganisms in that particular environment. So what about the human microbiome? Well, some of the most recent work suggests that your body consists of about 3 × 10⁻³ human cells. And it harbors about 3.8 × 10⁻³ bacterial cells. So you're just about half human. And this is actually an improvement because maybe 5, 6 years ago, they thought that there were many more bacterial cells than human cells. So, things are improving. The majority of those bacterial cells live in the colon. Now, I'm going to be talking a lot today about bacteria, but bacteria is not the only microbe that's living in our guts. There's also fungi, there are viruses, including phage, viruses that attack bacteria, as well as protozoans. All right, so let's talk about what we know about the microbiome from animal studies. And these are really some of the seminal studies that got people interested in the idea that the microbiome might influence the brain. And a lot of this work was done in germ-free mice. So these were mice that were raised without any exposure to microbes, a completely germ-free environment. They're mice in a bubble, essentially. And what they showed was that a microbiome is needed for the normal expression of many different behaviors. Probably the best studied is anxiety. So germ-free mice tend to have reduced anxiety levels compared to mice with a microbiome. Now, we're used to thinking of anxiety as being detrimental or problematic. But I think if you conceive of a mouse, a small prey organism, that being too brave, being unanxious, is actually maladaptive. The world out there is scary for a mouse. They should realize it. We also see that these mice have reduced levels of sociability, reduced recognition memory. In contrast, they actually move around more. They have increased locomotion and increased self-grooming behaviors, which is considered a sort of rodent proxy for repetitive behaviors. that you might see in conditions like autism spectrum conditions. Now, those changes in behavior are accompanied by changes in brain neurochemistry. So these animals tend to have lower levels of brain-derived neurotrophic factor, which is a compound that promotes neurogenesis, but increased levels of serotonin, of ACTH, and of cortisol, ACTH and cortisol playing important roles in hormonal responses to stress. And there are also more complicated changes in neuronal transcription and in neurotransmission. And what I find really fascinating is there appear to be certain windows of vulnerability. So this figure gives you kind of an overview of some of these mouse studies. If you have a germ-free mouse, if you have a normal mouse carrying a normal microbiome, they have increased levels of anxiety compared to a germ-free mouse, as I said. Now you can take the germ-free mouse and reintroduce a microbiome into it. If you reintroduce it relatively late, around 10 weeks of age, many of the aspects of neurochemistry I mentioned normalize, but the behavior does not. The behavior continues to be abnormal. Whereas if you colonize with a normal mouse microbiota around the time of weaning, you normalize both the neurochemistry, the hormonal responses, and the behavior itself. So it suggests there may be windows in early development when the brain is exquisitely sensitive to the impact of the microbiome. So what about in humans? So I feel that almost every week, new papers come out comparing individuals with psychiatric conditions of various types to healthy individuals who are typically developing. So it's been reported for schizophrenia, for autism, for major depressive disorder, for ADHD, and for anorexia nervosa. So that isn't necessarily super surprising, because when you think about it, having a psychiatric condition or being neurodiverse changes the way you interact with your world. So you can envision perhaps that someone with depression who's suffering from anhedonia might no longer have pleasure in food, and so they're going to change their diet. Children with autism often have very specific preferences about what food they want to eat, or their parents might put them on a special diet. So there are numerous factors that could change the microbiome after the fact, after the disorder or condition has already developed. But what's really interesting is that if you take microbes from people with psychiatric conditions and you put them into these germ-free rodents, you can actually induce some of the relevant behaviors. So one very influential study did this with microbiomes from children with autism. If you put them into germ-free animals, you see that the animals socialize less with their conspecifics, and their levels of anxiety change. And you don't see those changes if you take a microbiome from a child without ASD and put it into the rodents. This has been observed in microbiomes for schizophrenia and for depression and anxiety as well. So it suggests there may be a causal role for the microbes in brain development. Furthermore, what's also interesting is that genes linked to psychiatric risk may also impact development of the gut and the enteric nervous system. So the same genes that are increasing risk by affecting the brain directly may also be changing the environment that the microbiome colonizes, and they may be changing the way, or they may be affected by composition of the microbiome because they're in that same place. All right, so let's talk about the importance of infancy. So infancy is an extremely critical time, both in the development of the microbiome and in the development of the brain. And I'm going to start out talking about the microbiome. So this is data from a paper by Bakulich et al. This is out of Maria Gloria Dominguez-Bello's lab, looking at changes in the microbiome from birth through 2 years of life. Now, we think that the fetus is in an essentially sterile environment in the womb. But once labor begins and it's passing through the vaginal canal or being delivered via C-section, suddenly it's being exposed to microbes in the environment. So it's very quickly colonized. If you look at the figures here, this is looking at what we call alpha diversity. So I'm going to talk about two types of diversity this evening. Alpha diversity refers to the diversity within a particular sample. Here she's showing phylogenetic diversity. Probably the easiest kind of alpha diversity to understand is just observed species. The more species you have in your microbiome, the more diverse it is. But you can also consider things like the evolutionary relatedness of the microbiome. So that's this phylogenetic diversity. In an overly simplistic scenario, if you had 2 microbes in your gut and they were closely related, that would be less diverse than if you had 2 microbes that were distantly related. We can also talk about the evenness of the microbiome. So again, if you had 2 microbes in your gut and you had 50% of one and 50% of the other, that would be considered more diverse than if you had 90% of one and 10% of the other. But regardless of what kind of measure you use, we see that there are strong increases in diversity over the first 2 years of life, leveling out as the children become older. There are also major changes in the kind of bacteria that are living in the infant gut. Excuse me. So this here is a plot where each bar represents a month of life, and the colors represent different bacterial groups. So you can see that here, in the first month of life, or around birth, the community is really dominated by these proteobacteria. But they're very quickly replaced by other groups. So you'll notice, for example, there's an increase followed by a decrease in this light blue group. That's Bifidobacterium, which is a group that can feed on breast milk. So this is tied to changes in the infant's diet. And then you'll see as well that as the child ages, they cease breastfeeding, they start solid foods. You see increases in groups like the dark blue, this is Bacteroides, and red Faecalibacterium, which are more characteristic of an adult microbiome. Here's a slightly different way of looking at it. This is from a really large study out of some Scandinavian countries with over 900 children. This is the TEDdy study. They were looking at the microbiome and its potential role in diabetes. But what they did is they took all of their samples, and they had multiple samples from many of the children, and clustered them into groups. And that's what these circles here represent. And then the lines between circles represent how likely it is that a kid would move from one cluster to another when he had a different sample. So what they saw is that early life could be grouped into 3 phases. An early developmental phase where children's microbiome is changing rapidly, so they're very likely to move between clusters. And during this phase, all of the major phyla of bacteria are changing dramatically between samples. Then you come into a transitional phase where children still do move between clusters, but not as much as they did earlier on, and where only a few groups, including the proteobacteria and the Bacteroides, change significantly over time. And finally, infants around 3 years of age enter a stable phase where their microbiome essentially remains the same. You can see a few shifts here, but not many. So what's going on in the brain while all of this is going on in the gut? So I'm very fond of this particular figure from a review by Tebenkamp et al. And this is kind of a timeline of neurodevelopmental processes in the human brain. And the way they created it is they looked at gene expression, and they grouped the genes according to the developmental processes that they were involved with. So you can see that during the early embryonic period, as you might expect, very high levels of genes involved in cell proliferation, but these drop rapidly. Neurogenesis, so the birth of new neurons, primarily peaks around mid-gestation, but continues on all the way through the first year of life and into early childhood. And then you have synapse development and dendritic development, which increase throughout the pregnancy and continue to increase through that first year of life before leveling out. Now, using tools like magnetic resonance imaging, we can actually see sort of the large-scale results of these neurodevelopmental processes that we can't measure directly in a developing child. So this is the same child measured as a neonate, a 1-year-old, and a 2-year-old. These are different types of images. This one's called a T1 weighted, this is called T2 weighted. But the first thing that really should jump out is just the huge increase in size. So the brain increases by 100% in the first year of life, increases by another 15% in the second year, and sort of drops off thereafter. And in fact, by the time you're 4 years old, you're already at 96% of your adult brain volume. So that's not to say that later change isn't important, but the scale is completely different in these early years. Something else you may notice in this image, if we look at the T1 weighted, here at 1 year and 2 year, the white matter of the brain is actually white in the image. And this indicates that it's myelinated. The axons are surrounded by a sheath that increases the speed at which they can communicate. But in the neonate image, those areas are gray. And that's because at birth, the human brain is primarily unmyelinated. But as you can see, within that first year, it's already achieved a pattern of myelination that's very similar to what it will be in adulthood. Now, this isn't just about gross growth either. There's tremendous spatial and temporal complexity. So if you look at this figure here, these are changes in regional surface area over time. The top panel is the first year, the bottom panel is the second year. And the red areas are the areas that are expanding fastest. So you can see in the first year that primary sensory and motor areas are growing fastest, as you might expect, given the huge changes in an infant in terms of their ability to crawl and then to walk, increases in their ability to vocalize and vision. But it changes in the second year. In the second year, the pattern shifts. And now the bright red areas are in association cortices, those parts of the brain that are carrying out higher order, more complicated cognitive functions. And I'm going to point out this is inferior parietal cortex because we will come back to this region later, which has important roles in cognition, allocation of attention. In addition to the structural changes, there are important functional changes. So some of you may be familiar with functional MRI imaging in adults or older children where you're in a scanner, you're doing a task, you compare the brain to when it's not doing certain tasks, and you can see regions kind of light up, little spots of orange. We don't do that in babies because babies are usually asleep and you can't do a task with a sleeping person in the scanner. But what's interesting is that the brain is still communicating with itself when you're at rest and when you're asleep. So you can actually see networks involved in different functions are still temporally correlated with each other in a sleeping baby. This is a spring plot showing some of the major networks that have been identified first in adults. So these are things like primary visual networks, frontal parietal networks, which are involved in executive function tasks. And what you'll notice is each node is colored by the network that it's part of. And in the neonate, those colors are spread across from each other because they're not communicating strongly with each other yet. But as you look at the 1-year-old and the 2-year-old, you see the colors drawing together. So you're actually seeing the functional assembly of networks. And if you look at the overall within-network connectivity, you'll see that it increases for every one of these networks, with the notable exception of the sensory motor network. And you can see that in the bar plot as well. So we really wanted to ask the question, how are these things related to each other in infancy? What are the associations, if there are any, between the microbiome and brain development? And does it mirror what we see in the rodent studies? So our first study looked at the infant gut microbiome in relation to cognition. So we had 89 infants, 1 year of age, who donated fecal samples to us. It's easy to get poop from babies because it goes right there in the diaper, so they're convenient. They had their brains scanned at both 1 and 2 years of age, and they also underwent cognitive testing with an instrument called the Mullen Scales of Early Learning. And I'll say this was funded by the National Institutes of Health and the Foundation of Hope. And I also want to call out my colleagues and students who made a major contribution to this work. as I go through instead of saving it for the end. So this project was part of Alexander Carlson's dissertation research. He was an MD, PhD student in my lab. This sample was actually a subset of a much larger sample called the Early Brain Development Study, which is spearheaded by John Gilmore at UNC. Andrea Azcarate-Perillo is the head of the microbiome analysis core at UNC. Wei Gao is a colleague at Cedars-Sinai who did the resting state measurements, and Barbara Goldman headed up our cognitive assessments. And this has been published both in biological psychiatry, the cognitive data, and psychopharmacology for our resting state data. So the first thing we saw is that infants can be sorted into 3 clusters based on the composition of their 12-month microbiome. One of these groups is characterized by relatively high levels of Faecalibacterium, which you might remember is one of those adult typical microbes. One was characterized by relatively high levels of Bacteroides, which is more typical of earlier infancy, or middle infancy. And the third group, by relatively high levels of an unnamed genus in the family Ruminococci, and this is also a more adult-like microbe. Interestingly, if you run similar analyses in adults, with adult fecal samples, you also see 3 clusters, one with high levels of Bacteroides, and one with high levels of Faecalibacterium and Ruminococci. And the third one is something called Prevotella, which is usually associated with high-fiber diets, which is not what an infant is typically receiving. So it is possible that these groups represent a stable group membership, like we saw in the Teddy study. So it may be that these kids continue to be in these clusters as they age. We don't know that, but it's a possibility. The other possibility is that cluster is capturing maturation. So cluster 2 is the more infantile, the other 2 clusters are the more mature. And what we saw was that cognitive performance differed significantly by the cluster that the child was in, and was also associated with alpha diversity. So the figure here on your left is showing each child in each cluster and how they scored on the Mullen scales of early learning. So you can see that cluster 2 is the highest performing cluster. And I should say, none of these kids are cognitively delayed. They're all actually performing at a normal or above normal level. But the difference between group 1 and group 2 is a difference between a kid around the 70th percentile and a kid around the 90th percentile. So this is not a small difference. When we looked at the alpha diversity, we saw something surprising. Initially, we had hypothesized that children with high alpha diversity were going to perform better on our cognitive tests. And we based that in studies looking at the microbiome in relation to things like allergy and asthma, where it seemed like more diversity was actually protective. And there's an idea that more diversity enables a more robust microbiome, one that can reset itself when it's perturbed, one where there's functional redundancy of different pathways. But what we see is that the kids with lower diversity perform better on the cognitive tests. And we have a couple thoughts about that. One thought is it may be that the high diversity microbiomes are harboring pathogens that are not there in the lower diversity microbiome, and those have a negative impact on cognitive development. Another possibility is that this is related to maturation, somewhat like the clusters themselves, that maybe having a more protracted period for development of the microbiome is actually beneficial to brain development. A third possibility is that the high diversity microbiomes may have more competition between the different groups for resources. So the beneficial bacteria may simply be outcompeted or not have enough resources to fully exert beneficial effects. So you may be asking, okay then, so what is different between these clusters? How do they function differently? So what we did is we used the genetic sequence to predict what metabolic pathways, what functions these different microbial communities might be performing. In this figure, 0 represents cluster 1, so that's serving as the baseline. Blue represents cluster 2, red is cluster 3. I'm going to focus on cluster 2 because they were the high-performing cluster. So the first thing we noticed is that this group had more metabolic pathways involved in processing vitamins and metabolic factors. So things like folate, which is very important for brain development, biotin, lipoic acid. So this kind of fits in with that hypothesis that this cluster is performing positive things for the host. It's giving them more energy, more nutrients, more vitamins. Cluster 2 had less pathways involved in bacterial motility, and sporulation, and bacterial chemotaxis, and flagellar assembly. These are all things that microbes do when they're gearing up to make trouble. So that would be in keeping with the hypothesis that there are pathobionts in the poorer performing groups of kids. You may also be asking, okay, so what are the neural circuits that the microbes might be impacting? We looked at this with our resting state connectivity data, and we did it two ways. The first thing we did is we wanted to look at those functional networks I showed you in the spring plot. So what we did is we picked a node in the brain to represent each network, and then tried to identify other areas of the brain that were connected to it, and where that connectivity was related to alpha diversity. And what we found was that functional connectivity, between the supplementary motor area and left inferior parietal cortex correlated with alpha diversity, and moreover, it also correlated with cognitive performance at 2 years of age. So this looked like a neural circuit that might actually be mediating the impact of the microbiome on cognition. So I pointed you to that bright red IPL before. So that was that area that was growing really fast in the second year of life. And as I said, it's involved in top-down attentional allocation, social cognition, and language. And you can kind of visualize how all of those skills would be really important for a child as it's learning. Oh, and I should point out the node that we used here represents the sensory motor network, which again makes sense when you think about how infants learn about the world. They're learning about it through their senses, through manipulating objects, through sticking them in their mouth, through what they see and hear. We also saw that functional connectivity between the anterior cingulate cortex and the right anterior insula correlated with alpha diversity. Now, the anterior cingulate was chosen as the node for the salience network, which is involved in sort of processing incoming information and deciding what other networks ought to be turned on or turned off. But it also has a key role in emotional regulation, as does the insula. So this was really in keeping with the rodent studies suggesting a particular influence and anxiety-related circuits. Which is why we took our second approach, was to look at a particular structure called the amygdala, which has an important role in processing threat, and see what other regions what it was connected to and where that connectivity correlated with alpha diversity. And what we found was a relationship with this cluster you see in blue here, which overlaps with the thalamus, and in particular with the paraventricular nucleus of the thalamus, which we know is strongly connected to the amygdala, and we know that that particular circuit plays a key role in fear processing and negative emotionality. So again, this suggests that the gut microbiome in 1-year-old infants may be related to the processing of negative emotions like fear. And the next study I'm going to show you set out to explicitly test that hypothesis, because in this study, we didn't have any measures of fear behavior. All right, so I'm going to tell you about the infant gut microbiome and anxiety-related circuits. For this study, we had kids come in for 2 visits. Thank you very much, I appreciate that. So they came in at around 2 weeks of age and around 1 year of age. We got fecal samples at both visits. We collected saliva, both before and after a heel lance for blood collection. The blood collection will allow us to look at certain signaling molecules, and then the saliva will allow us to look at cortisol responses to this stressor. And they got an MRI of the brain. At 1 year, we also did behavioral testing for fear reactivity. And we used 2 tests, the strange situation and the mask task, which I'll show you videos of in a minute. And we also did cardiac recording while the children were undergoing this testing to see how their heart rate responded. Some familiar faces here. Andrea did our microbiome again. Martin Steiner oversaw our MRI analyses. Alex, again, this was part of his dissertation. Kieran Gruen advised us on the cortisol. Kathy Proper advised us on the behavioral testing. And then Samuel Rosen is a graduate student who carried out the hormonal statistical analyses. All right, let's see if the videos work. Moving. I hear her, but I don't see her this time. Oh, there she goes. All right, so in this strange situation, the mom and a research assistant, who the child already knows, bring the child into a new space. They get to sit down there and play with all these new toys. They usually like that a lot. Well, mom sits and reads a magazine in a chair. But after a few minutes, a stranger enters the space. This is a research assistant that they have not met before. There he is, so that's Alex. And you can see as soon as he comes in that the little girl kind of freezes with her arms up in the air. And she basically stays like this for about 30 seconds. She is very suspicious of him. And this is actually pretty unusual. Children are usually less bothered by the stranger. Many of them actually will approach the stranger and try to engage them in playing with the toys. But she is having none of it. Our second task is the mask task. It's very much what it sounds like. The child is in a high chair, and behind that curtain is our research assistant. And they're going to try on a Halloween mask. Come out from behind the curtain and say the baby's name a couple times. And depending on how the baby reacts, they can see between 1 and 4 masks. So we'll wait for a minute for our first mask to come out. So, as you can see, he doesn't like the mask. And I've obviously chosen these videos because they are some of the most extreme responses we saw. We do try to make this a warm experience for the families, so we never let a child cry for more than 10 seconds at a time. If they have a really strong reaction on the first mask, we don't show them the second one. They've already shown that they're a highly reactive kid. But one thing I find really interesting about him is although he has a really strong reaction, as soon as that mask is gone, he settles himself right back down. So he also appears to have quite good kind of cognitive control or emotional regulation for his age. So I'm going to show you the stress response test first, although I don't have cool videos of that. But when 1-month-old children experience a heel lance or an inoculation, they have a very strong cortisol response. And you can see that here. So this is baseline before the heel lance, 20 minutes after, and then 40 minutes after. So most of these kids show a strong increase in cortisol, and then they recover, getting back to where they started by the 40-minute period. So we looked at our microbiome features in relation to the change from 0 to 20 minutes, how strong the initial response was. And what we saw was that alpha diversity, so again, the overall diversity in the samples, was related to hormonal responses, such that kids with more alpha diversity had a stronger cortisol response. And we know from other studies that kids with extremely strong responses at this age may be at risk for later anxiety disorders and depression. We also found the diversity of the 1-month microbiome predicted fear reactivity in the mask task when it was administered a year later, which I think is really interesting. We did something a little different with our alpha diversity measures here. We did something called a principal component analysis to kind of pull patterns out of them. And the first principal component seems to represent richness, so just the number of different species that are there. Whereas the second component really seems to be capturing evenness. And what we saw was that kids who had more even and less rich microbiomes had less pronounced responses in the fear task. We also saw, I'll draw your attention down here to these things labeled WUPC1 and WPC2. This is a way to look at a different type of diversity, beta diversity. And this is the same kind of thing we were looking at in the clusters, but here we're doing it in a way where instead of dividing up the kids into groups, we can look at it as a continuous measure. So in this case, we see a particular association with PC2. Kids in that group have high levels of Enterobacteriaceae and Bifidobacterium and low levels of Veillonella, and they're showing more fear. We also found the composition of the 12-month microbiome was associated with concurrent fear reactivity at the same time. So this is another one of the column plots, but here each column represents a kid. The colors, again, represent different bacterial groups. So this gives you a sense of what those principal coordinates are capturing. So this is organized by the first principal coordinate, and you can see that this gray group, this is Bacteroides, seems to kind of track with that PC. So in this case, what we see is infants with high levels of Bacteroides and who also have low levels of some other groups like Veillonella, which is the medium blue, Dialister, Bifidobacterium, which is the dark blue, and Lactobacillus showed decreased fear reactivity. We also found that features of the infant gut microbiome are associated with volumes of the amygdala and medial prefrontal cortex, so two regions that play an important role in fear processing. So at 1 month, weighted unifrac PC1, that's just what we're calling them, had strong positive associations with Bifidobacterium and Streptococcus, strong negative associations with Bacteroides, and it is related to 1 month, it's related to 1 year medial prefrontal cortex volumes. So again, predicting what's happening later on. And most exciting, we found that the same principal coordinate that was associated with fear reactivity at 12 months also was associated with amygdala volumes at the same time. So again, that suggested a potential structural change mediating the behavioral differences. All right, so I'll stop talking about my work and go out to kind of the bigger picture. So if all this is true, how does it happen? How does the microbiome actually talk to the brain? Well, there are a couple of different ways. One, microbes in the gut can actually change the function of enteroendocrine cells in the gut. These are cells that produce most of the body's serotonin, which is an important mood regulating chemical. So they can interact with those cells and change the production of serotonin. They interact with the immune system, both the intestinal immune system and the systemic immune system, and they can change the production of both pro-inflammatory and anti-inflammatory cytokines. Those can affect brain development. We've been really interested in the fact that molecules make chemicals as part of their metabolic processes. And some of those chemicals are neuroactive. They make classic neurotransmitters like dopamine, GABA, glutamine. They also make precursors of serotonin. It's probably unlikely that those compounds are traveling through the whole circulation to get to the brain, but they could be signaling through the enteric nervous system, which has a direct route to the brain through the vagus nerve. And in fact, some of the rodent studies suggest that if you sever the vagus nerve, you actually stop the communication between the microbiome and the brain. Another hypothesis that's been proposed is the so-called leaky gut hypothesis. So in this model, you have a healthy central nervous system and a healthy gut. Okay, they have close junctions with each other, so the bacteria remain sequestered from the rest of the circulation. However, in the context of stress or disease or gut dysfunction, those barriers can open up, and now you have pathogenic bacteria from the gut entering the circulation. I know I'm running out of time, so I do still want to talk a bit about clinical implications. So if we think about microbes, microbes can be factories that produce drugs, they can act as drugs, they can modulate the action of drugs, nutrients, and environmental chemicals, they can deliver drugs, and they can be a target that's killed by them. And I want to just very briefly talk about all those. So microbes make drugs. Compactin, the grandfather of the entire drug class of statins. Japanese microbiologists found it in mold in a rice shop in Kyoto. Sirolimus, which is an immune suppressant that is administered after organ transplant. It was found in soil from Easter Island. Doxorubicin, it's an anti-tumor therapy, found in soil around Castel del Monte, a 13th century castle in Italy. So I just think it's fascinating that these things have health effects. Most people are probably familiar with probiotics, so live microorganisms that when you administer them can confer a health benefit to the host. You may also see talk now about prebiotics, so food ingredients that can induce the growth or activity of beneficial microorganisms, or symbiotics, which include both in the same package. You may also have heard of fecal transplants. So this is where you actually wipe out the person's native microbiome with antibiotics and replace it with a new microbiome from a donor. Microbiomes also process drugs. Since I'm running out of time, I'm just going to focus actually on the bottom here. So these are several psychiatric medications that we know are processed by the microbiome and that could impact their efficacy and their side effects. So risperidone, a new generation antipsychotic, levodopa, which is a dopamine precursor, nitrazepam and clonazepam, hypnotics, and methamphetamine, a psychostimulant. They also process environmental chemicals. Some, like methylmercury, they actually make them less toxic. Others, like nitrobenzene, they can make more toxic. And they process nutrients. One of the major roles of the microbiome is to perform caloric salvage, to actually get energy out of food that you can't get yourself. And we know that caloric restriction has huge effects on brain development. There are also potential associations between the microbiome and fatty acids, and iron, zinc, choline, and B vitamins, which goes back to what I showed you earlier with those different metabolic capacities in our different clusters. Microbes can deliver drugs. I think this is really cool, although I don't do it myself. You could actually genetically engineer microbes and introduce them into an organism. No one's done it for psychiatric conditions yet, but this is a pretty early study showing that if you put in a microbe that had been engineered to create an anti-inflammatory cytokine, it actually helped control inflammation and colitis in an animal model. And finally, microbes can be killed by drugs. So if we could figure out who the unbeneficial microbes are, we could take them out, possibly with antibiotics or with bacteriocins. Antibiotics tend to have broad spectrum effects. Bacteriocins are compounds that those warring microbes, the competitive microbes we talked about earlier, produce to kill off closely related strains in the same niche. And there's interest in using bacteriophage, so viruses that actually use bacteria to reproduce and often kill them in the process. So either naturally occurring or genetically engineered phage could be used to control microbes. So in conclusion, certain features of the gut microbiome are associated with behavior, brain structure, and function. The composition of the microbiome may be a factor we can manipulate to improve long-term mental health. Microbes can be manipulated in multiple different ways, but the mechanisms by which specific microbes or microbial communities alter infant brain development remains to be deciphered, and that's the focus of my work going forward. So back to our first questions. Who thinks that manipulation of the gut microbiota is a promising strategy for supporting cognitive development? Oh, yay, there are more hands. And is manipulation of the gut microbiota a promising strategy for treating or preventing psychiatric disorders? Okay, we're not as sure about that one, but we've got more hands. So thank you all very much. I do have an analogous slide for the many, many people in my lab at MSU and my collaborators back at UNC. You can see a picture of our beautiful building and my wonderful graduate students, postdoctoral fellows, and undergraduate volunteers. So I'm very lucky, and I'm very lucky to have had families who were interested and excited to participate in this research. All right. Yes, the lady in the beautiful sweater. All right, so the question was, do the clusters that the children fall into relate to what their families, their siblings, or their parents have? I think that's a wonderful question. We haven't asked it directly. We don't have mom or dad's or siblings' microbiomes. But I will say that we do see that having older siblings affects the microbiome at this age. So as you might expect, if you have older siblings, you have a more diverse microbiome because they are bringing microbes in from more diverse environments. Yes, second question.
So my second question is, are you finding that children who are in NICU or who are breastfed longer have delays in that biodiversity increase?
All right, so the question is, does being admitted to the NICU or being breastfed affect changes in diversity over time? So we did not find that being in the NICU, and we just said if you're in the NICU over 24 hours or not, impacted either cluster membership or alpha diversity. But it's not a huge sample size. It's about 89 children in that first study. So actually my MD PhD student is very interested in NICU babies, and that's what he's doing now in his residency research. We do see that breastfeeding impacts these things. Breastfeeding is one of the largest components for microbiome diversity in early life. Although even it only explains about 5% of the variance between kids. So it's sort of one of the biggest ones we know of, but there's a whole lot of variation that is not explained. And typically breastfed infants will have less diverse, more infantile microbiomes. I'll also say cluster 2, the high-performing cluster, is more likely to be breastfed, is more likely to be vaginally delivered. The relationship is there if you control for those factors, but those are characteristics of that cluster. Yes?
Kind of following up on that, I was wondering if in this study you kept track of the diets of the kids to see if that varied at all, if that could have affected your results.
Yeah, so the question, does the diet of the child impact the microbiome? So in the first study, we were piggybacking on an existing cohort. So we had data on breastfeeding versus formula feeding, but we didn't have much else. In our anxiety study, we've taken much more detailed dietary questionnaires, both at 6 months and at 1 year. We didn't see any relationships in the group of kids I showed you today, but that study actually was a 2-phase study. So this was phase 1. We're now analyzing the data from the second phase, which is a larger group of kids. And they're different in many, many different ways. But one thing that is different is we do see some relationships with, let me think about some of them, with the servings of formula that they received at 6 months of age, when they ceased getting breast milk, whether they got French fries when they were 1 month old. And I suspect that all of these things are kind of tapping into a more general construct about being able to do extended breastfeeding and being concerned about the diet that you're giving your infant. Yes? Yes, so the question is, has anyone tried using probiotics to treat psychiatric disorders? And the answer is absolutely yes. The best studied disorder is depression, and there are probably 20 to 30 studies that have looked at it. And it's about 50-50 whether they show a positive effect or not. So not necessarily super compelling. There's sort of a handful of studies on autism, and again, kind of 50-50. I think the most interesting one is actually a study that was administering the probiotics early on, and their goal was to look, I think it was at allergy and related outcomes later. But they also looked at neurodevelopmental disorders, and they grouped autism and ADHD together because they hadn't powered the study to look at psychiatric conditions. They'd powered it for their other outcomes. But that suggested that the early probiotic treatment actually reduced risk later on. And I think that's interesting. And one of the reasons I'm interested in infants is I think that the microbiome may be more manipulatable at that time, as well as the fact that you could be preventing an abnormal trajectory in brain development, as opposed to trying to correct it after it's already happened and you're way down the pathway. One interesting thing about probiotics for treatment, there's a big study a couple years ago suggesting that a lot of people don't get colonized by probiotics. So there are some people who have guts that are colonizable, that are welcoming, but most people don't. So that may be one of the problems with the studies as well, that a lot of people just simply didn't get any benefit because they didn't colonize. Oh, so I'll do over here. Yes, so the question is, did we control for prenatal cortisol exposure in our study of anxiety-related circuits? And the answer is no, because we didn't have maternal measures of cortisol reactivity. One of my colleagues at UNC is interested in the pregnancy microbiome and how it impacts maternal mood and vulnerability for postnatal depression. So she is collecting some data that would address that question. We do see in our phase 2 study, so we don't see any relationships between life stresses reported by the mom in the first year of life and how infants respond, but the mother's own trait anxiety does predict they're responding on the task. So moms who report being more anxious themselves, their kids respond more strongly. Yes? So the question was, what can you do to restore a leaky gut? And I will confess, I don't actually know exactly what's going on in that research area. I'm sure it is an active area of research. I'm sorry I had to rush through it. The figure I showed you of the engineered microbes and how they're impacting inflammation, I would make a guess that those same kind of compounds would also be useful in terms of repairing leaky gut. But I am speculating there. It's not my area. All right, and there's another question over here. So the question is, do antibiotics early on disrupt brain development? So we have not seen strong associations. Well, now I'm thinking. We haven't seen strong associations between antibiotic use in our cohorts and their microbiome levels. We haven't directly looked at whether they've impacted the brain phenotypes. That would be a slightly different question. I think part of that may be that, particularly in the second study, we required that until at least the first visit, the kids had to be breastfed, they had to be antibiotic naive, the mom had to have not received any antibiotics for the last 2 weeks of pregnancy. They also had to be term and normal birth weight. Some of the kids did get exposed to antibiotics after the first visit, before the second one, but it may be that we just had a group that didn't have as much exposure as some other samples might. Although jumping back to one of the very first figures I showed you from Maria Gloria Dominguez-Bello's group, so her work suggests that antibiotics do disrupt the developmental pattern of the microbiome, but eventually kids do catch back up. Yeah, so the question is, if infancy is a time of vulnerability for microbiome effects, could there be a second developmental window, specifically adolescence, where they might also be impacting brain development? So there are groups investigating that, and in fact, I'm involved with one that's looking at not adolescence, but mid-childhood microbiome levels. And you do continue to see some change in the microbiome composition from late childhood through adolescence into adulthood. You know, a little like the brain phenotypes, you know, it's not as marked as it is in those earlier ages, but change is still happening.
Well, why don't we all take a moment to thank Dr. Nickmeyer for that great presentation. I want to thank our hosts again, Calvin University, for the use of this beautiful facility. Also, thank you to co-sponsors Spectrum Health and Michigan State University College of Human Medicine. Thank you, Arlene, for the hospitality. Thank you, Terry Crum, also for all the work you did to make this event possible. And hopefully we'll see you again next year. Have a great night.